us start today's session, and I am privileged and very happy to introduce Dr. Alban Sigamani. Dr. Alban completed both MBBS and MD in pharmacology from St. John's Medical College, Bengaluru, and served as faculty from November 4 to August 2015. Dr. Alban Sigamani is currently heading the clinical research at Narayana Health, Bengaluru. Since August 2015, he handles clinical research projects of entire NH Group. He leads a team of 50 members to coordinate multiple clinical trials and clinical research projects across different departments and various units across India. He is an expert in regulatory affairs, research methods, and biostatistics, and provides support for budget and contracts in clinical trials. He trains, mentors, and guides students doing fellowship, PhD, and clinical residency. He nurtures a clinical research culture and network with different stakeholders to increase research output in his institution. He is consultant for medtech innovation entrepreneurs. He provides clinical immersion for medtech innovators and creates a platform for entrepreneurs in healthcare startup industry. Dr. Alban teaches international MPH courses offered by Euclid University on the learning management software. He is an international faculty and associate professor in public health since April 2013 in Euclid University. Dr. Alban Sigamani is a teacher, clinical researcher, clinical pharmacologist, clinical trial expert, and clinical epidemiologist. He is also interested in history and philosophy of research, which is very interesting. Now, over to you, Dr. Alban.
Good morning, everybody, and I hope you can hear me well. Thank you so much, Vidya, for that kind introduction, and also thank you, the organizers, for hosting such an event, specifically trying to inculcate interest in drug discovery and also applying their young minds into challenges of health. And it's my privilege to be giving a lecture today, and also trying to see how we can, you know, address, give some thoughts, food for thoughts for our young hackathon participants, and to see how we will be able to gather, uh, at least make some initial stepping stones with regards to solving some of the big challenges of health that is that has been affecting humankind for a long period of time. So, so to go straight off without much any delay, I hope the audio is clear. Just making a last minute check with you.
Yeah, I think everything is fine. You can start.
Okay, thank you. Okay, so today's topic is about scope of targeted therapies, and it's a little, it's a pretty large topic which you can have an entire workshop on. And so I'm just going to kind of give you an oversight about when we mean by targeted drug therapies, what does it mean? And I'll kind of take you a little bit into understanding the need, or where did this kind of whole aspect of targeted therapies come in, and how it has actually influenced the modern medicine in general. And we always think about targeted therapies only in lines of the newer anti-monoclonal antibodies or newer genetic therapies that are being used in cancer or in any other kind of chronic diseases. But the concept of targeted therapies actually revolutionized the way human understanding of disease started to happen. So, if you really ask me what is the scope of targeted therapies, it has started off with infectious diseases. When we first identified the target as a bacteria or a microorganism that is causing an infection, and then that was leading to disease, and there was a need for us to target it. So that was kind of a starting point of targeted therapies. Then the whole concept of non-communicable diseases came into picture, and they said that they couldn't give it a different terminology, so they just used non-communicable diseases. And so whatever is not able to spread from one person to another person, they said it will all be considered as non-communicable diseases. So that's what the word non-communicable means. But over time, people started understanding that infectious diseases were not just something which happens as an acute event. You can live your entire life with an infection. HIV being one of them, which kind of changed the whole thinking process around infection. They thought that you get an infection, you either have it for some time. If you are unfortunate, you die. Otherwise, if you're fortunate, you recover and then you don't get that infection again, or you don't have the infection in you for some time. But the concept of that changed when HIV came into picture, and you have people who used to initially die now have started surviving, and you have long-term survivors with infection. And tuberculosis was another big disease that also brought in this whole concept of chronic infectious diseases, just like leprosy. But the understanding of those diseases were kind of little vague at that point in time. It took many hundreds of years compared to HIV, which is more recent. I would say that most of us who are participating in this today's webinar would kind of find it kind of happened sometime in our lifetime. So this has also led to the concept that non-communicable diseases can also be chronic. You don't get diabetes for a day, you don't get hypertension for a day, you don't get heart disease for a day. It kind of comes in, and then you have to change your whole lifestyle, and you need to kind of live with it for a long period of time. So the scope, I would say, is kind of it started off with infectious diseases, has now spread across all forms of diseases. But before we go into disease, let's look at what is health as such. The definition of health, I'm sure many of you all will know, which WHO made, as when they were forming the charter, and they said they need to have a definition for health because they're standing for health. So it's a World Health Organization. So they said that health is a state of complete physical, mental, and social well-being. And it's not merely the absence of disease or infirmity. So sometimes we think health is the opposite of disease. And that is kind of a concept that came in because of targeted therapies. We felt that somebody is got a disease, that means he's not healthy. And if he does not have a disease, that means he's healthy. But then WHO said it's not just a disease. It's just not that you're having a disease or not having a disease. If you're not happy with your physical activities, your mental being, your social well-being, even that is considered to be unhealthy for you. And rightly so, because later on I think this concept was there for some time, but more defined by epidemiologists, which said that you have actually a disease as a triangle. And the three parts of the triangle is the environment, the host, and the agent. And so the agent is what is causing the disease, and you have the host that is getting the disease, and always you have the environment which is kind of bringing about this disease. And then the extent of damage to the host by the disease, you see a large population of people who are kind of remaining unaffected, and a smaller group of people actually getting affected and having succumbing to the disease. So it's the severity of the environment, the more focus of the environment, the more prevalent the pathogen, you start to have more concentrated people with the disease. And this is what we are seeing very much commonly now with the coronavirus that's going on. the prevalence of the pathogen is kind of spread so much. That's why you're getting a large number of people with the disease. And the environment is kind of making it happen because we've been living in crowded places. Our environments are not clean. And the host also is contributing, because this particular virus would not have been such a major problem if we were actually healthy. If you look at the definition of health, and if we had that kind of health with us, which is physical, social, and mental well-being, we wouldn't be succumbing to this particular virus. So if you really look at it, the play of corona is kind of clearly showing us what is happening with this disease triangle, and everything is interplaying in it. So it's important that we keep all these components under control. The lesser the susceptibility of the host, the reduced whatever virulence of the pathogen is, the reduced amount of infection that you can have. The cleaner the environment that you have, it supports the host from having the. Now, why is this important? We sometimes forget about this when we get into the whole concept of targeted therapy, and we think that everything can be solved by like switching off something. You find something that is going wrong and you kind of switch it off, but you kind of forget. And I think that's what I wanted to keep everybody's in mind here, that you should kind of have a kind of a wholesome view or a systematic view of the whole problem. It's not just that virus. So you can't solve the corona epidemic by just fixing the virus. You can't just have a antiviral, or you can't have a vaccine that fixes the virus without improving the person's health or changes in the environment. So it's kind of become a challenge that we need to deal with. And so, targeted therapies, when we think about, we need to keep this in mind. It's not just finding a cause for an effect. Now, epidemiologically speaking, from research perspective, we are always constantly grappling with this whole concept of causation and association, which is also important. Now, when we say we are doing targeted therapies, we find a target that we want to change. So we're thinking that that is the cause, but is it an associated factor or is it a causative factor? This differentiation is very important. We all know the classical thing about exposure leads to outcome, and you have a large exposed group and a large outcome group, but only a few percentage of them actually have both sitting together. So it always brings in a lot of confusion. There used to be a time when cigarette smoking was considered to be a healthy habit for people to have, and that was because there was a large people who were getting exposed, but that was not kind of seen. Exposed people were not having outcome. A lot of people were having lung cancer even without smoking, and a lot of people were having heart disease without smoking. So people found it difficult to kind of understand why exposure and outcome. So this concept of exposure and outcome is always a constant debate. And when we talk about targeted therapies, we are only able to kind of address this middle section where you can find a temporal relationship between the exposure and the outcome. We've seen that the exposure has happened and then the outcome has happened. And that's always a small section of the population. And this is also a big challenge. When we're thinking of a particular targeted therapy, can we actually wipe out the outcome? It's difficult. If I take off the exposure, will I be able to completely wipe out the outcome? It's difficult. People could have done that with cigarette smoking. They could have done that with alcohol. They could have done that with many conditions for that matter. But very few people have been successful. I think the only success story that we can talk about is the smallpox success. We were able to kind of wipe out smallpox and control it completely. And so now we don't have smallpox in our hand. To that extent, polio seems to be reaching that kind of a thing. So, two diseases out of a million diseases that is affecting the billion people is what we've been able to achieve over the last many hundreds of years of our existence. So you can understand the limitations that you have. So again, why I'm bringing this about is just you should not have this thinking that, oh, I have found the target, I can do something about the target and then I think I will wipe out the outcome. It's not going to happen that way. You will only be addressing a small component of the whole thing. And that is why it is also important to understand this relationship between causation and association. So you'll see that a lot of exposures are actually just associated with the outcome. And if you remember from the previous slide, there are other factors which actually lead to that kind of exposure leading to the outcome. Another problem that we deal with is about correlation. This is a kind of a cartoon which just shows that number of people who drowned by falling into a pool correlates with the films that Nicolas Cage appeared. Nicolas Cage is a Hollywood actor. Many of them may know. So somebody just sat down and correlated these two. So the point here, friends, is that when we look at correlations, we have to be very careful about what we are correlating it and what is the clinical or the biological explanation to it. What does it have with Nicolas Cage appearing in a movie and people who drown? It has no sense. But if you actually sit down and correlate it, you see how beautifully it is correlating. So what I'm trying to say here is that just by finding a signal, you find a signal for a particular disease and you find that signal to be very strong, you should not think that that signal is giving you the answer. And this is also important when it comes to drug discovery in general, and a big fallacy that sometimes happens. And sometimes when we miss out on this, we have major disasters that happens with drug discovery. You go by a particular a particular signal, and you said that, oh, I found the signal to be very strong, and I kind of, so if you do not have a biological reasoning behind it, it makes no sense. Now, you can draw beautiful graph like this, but it will make no sense at all. So I was already talking to you about lung cancer, and so people talked about, when people are talking about lung cancer, there was a big debate that lung cancer is more seen because of low socioeconomic status. People living in poor economic backgrounds are getting lung cancer, and nothing to do with the tobacco smoking. In fact, all the CEOs of all tobacco industry people said tobacco smoking was safe, and it took a long time for them to really make the difference. And if you say in the last 10-15 years only, when really there's been a movement about tobacco smoking and causing cancer. And the reason for that is because you have such a lot of biology effect that so the point here I'm trying to say is that biology is very confounding. It is because it is not like a computer. It's a computer is, it's driven by a software and it has certain hardware. So you have a hardware problem, it's, what the problem is. It may be an electrical problem, it may be temperature regulation problem, or it could be some particular physical damage to something. If it's a software, where the coding is could have gone wrong, you could find it, fix it, and do it. But in biology, it doesn't happen that way. Biology is constantly evolving. Even in our own bodies, it's evolving in different times and different stages. Even now, as we are adjusting to working from home, spending more time with family, and being indoors and all of that, I'm sure our biology is changing quite a bit. And I'm sure somebody will be interested in studying that and understanding, has it reduced chronic diseases, has it reduced certain other aspects of it. So a lot of effects that will happen. So it is this confounding factor is what is also an important, I would say a fallacy when it comes to looking at targeted therapy. So cause and effect, environmental influences, the biology. So if you see, you're dealing with a very unstable system, and it's not a system that you can grapple at one, you can't sit in one place and you can't put every component of it together and say that I found a solution. So it is always going to leak out in some place or the other. So the best thing you can only do is try to address what is the bigger problem and try to reduce it to an extent. You can't kind of wipe out a disease. That's one of the reasons why, if you see tuberculosis, so far they've been trying to do eradication programs. Now it's gone back to a control program. Leprosy, to an extent, they've been able to achieve eradication, but it's still very it's still present in certain pockets, and people do live with that as a chronic infection. So we don't know about COVID, what's going to happen. So I don't know whether we'll be able to eradicate COVID completely, but it's going to remain in certain pockets. Hopefully, we'll be better adapted to, because our biology will change, and we can be better adapted to getting infected with COVID. So this is how the whole health and disease kind of parameter works, and it's very important for somebody thinking in this lines of finding a new treatment or a few new diagnostic or trying to find a solution for a health problem should always understand that health has multiple components to it. It is not just merely removing an infection. It is not in just by a cause and an effect. You can't just have one cause and eliminate the cause and see that effect will go off. And when you look for signals, you should be very careful about signals which are not biologically explainable. And even if you find a biological explainable thing, there will always be confounders which you need to adjust for. So, in biology, in drug discovery, the aspect of scientific reasoning is very, very important. And I think each of y'all getting into this journey should understand that very strongly. Just to give you another example about confounding, see, the bacteria for tuberculosis was kind of identified way back in 1870, 1800. Then they identified a tuberculin test early in 1890. And it only, the almost 50 years, 60 years later is when they had a vaccination that was developed. And then antibiotics came on. Once the vaccination was developed, they understood the bacteria a little better, so they were able to take the bacteria into a laboratory to start doing antibiotics. It was only in these years that you started having certain antibiotics which started bringing down the mortality. So if you ask me, mortality used to be so high, it started coming down only from 1860 to almost 100 years it took for us to bring some kind of a sense to tuberculosis. So this is a timeline that shows the events in drug discovery. Like I said, people have been playing around with chemistry for a long period of time. We can only go by whatever is recorded in literature. So if you look at Indian literature, probably it will go back into BC that people have been, experimenting with natural compounds and basically they've been doing chemistry. And only later on, when they were able to kind of develop analytical chemistry. See, previously chemistry was like it was a science that they could not define, and they only used to see observation. So it used to say that, okay, you put chemical A and chemical B, you'll get a color out, you'll get a reaction. But they were not able to quantify it. Only when they were able to develop analytical chemistry did it really make a quantum leap as far as drug discovery is concerned. And the addition of analytical chemistry into the discovery of biological pathogens, which happened in thing, actually led to first pharmacology, which started with receptors and identifying. So identifying a particular receptor, identifying a target, and we started having drugs coming out, and then that led to advances in biology. And so now we are in this phase where we're looking at targeted therapy for cancer treatments, targeted therapies for HIV, targeted therapies for various chronic conditions that is kind of coming in. And it is a lot of that biology understanding kind of helped when we were able to completely map the human genome, which started off in 2000 and kind of got completed in 2003. So only when we really understood our entire genome, did we figure out that we cannot really get a grapple of the entire biology immediately. So it's going to take many, many, many more years for us to really understand. But as nature always does, it does it in phases. But if you look at it, the time period that used to happen for each phase will start to come down, like 1815 to 1865, then started reducing, and now you have discoveries happening every day. The internet revolution has actually made it possible for people to start seeing discoveries and changes happening every minute. I think we keep track of our LinkedIn, or you keep track of your uh, any of the social media or FaceTime, Facebook, anything you keep, you'll be able to start seeing discoveries happening more rapidly. So things have, like, even as far as Corona is concerned, from March to in the last 6 months, I think there's been a huge amount of data and huge amount of discoveries that have happened in understanding biology of not only of the virus, but also in the biology of the human being's immunology. A lot of understanding has gone into that also in this very short period of time. Another example, all of you all know that acute myocardial infarction is a heart attack. And basically what happens, it blocks, you have a thrombus which goes and sits inside a artery, and the artery blocks, and you don't have blood supply to the cardiac muscle, and that's how it dies. So if you look at these timelines, fibrin was identified way back in 1666, and plasminogen was identified in 1941. 1933, streptokinase as a molecule was identified, and plasminogen getting converted into plasmin was only in 1944. So you see, 1941 to 1944. Then the last step of fibrin to fibrin split product, 1666 is our understanding, but we didn't know what to do here. So only when, see, if this was something that was happening in now in the current age because of internet, probably all of this would have happened in a single day. But see the timeline it took. So if you really ask me, streptokinase treatment really became effective in treating acute MI only in 1970s, way down in 1970s. Even after all the discovery was done, it took almost 55 years for them to find the evidence that it used to work. So this is another angle I wanted to bring into the whole picture, is that acceptance of discovery, especially in biology and especially in the clinical medicine science, takes time. You can't have a discovery and you can't think that it will kind of become part of practice tomorrow. You can't identify a target and find a target therapy for the target and think it will kind of change things from tomorrow or the next 1 year or something. It takes time. It would have been, it just contracted significantly, but it takes time because the understanding of it in the clinical world, it needs more time. So this is just another example to be cautious, that don't think that, okay, you have a discovery today, that it will become a drug tomorrow. So it's going to take time, and it's probably not going to take this long, but it's going to definitely take a period of 5 to 10 years time. So why that has happened? What is actually different that has happened? So now, if you look at it from a drug discovery point of it, you have the combination of biology, the pharmacology, chemistry, and then when the genomic project was completed, the genes made us understand the biology a little better, and then we were able to identify targets, and we came out with pharmacology. And for that, we identified chemistry. So this is how drug discovery is happening. And what is kind of core in the middle of all of this is technology. And by increasing our computer capacities, our ability to process large amount of data, I think nowadays the computers are able to process terabytes, or I don't know if there is something bigger than that, gigabytes or something like that, kind of amount of data it kind of produces. So it's capable of functioning at the level of 1 million human being brains, they say. And our mobile phones is equivalent of our 10 human brains, they say. So technology has really come down very, very big, and it's contributing in this whole area of drug discovery. So you have that in your hand. So it's a very powerful tool, but you have to be careful that the danger that is there is that all of this is only giving you an understanding of the problem. It is not going to find you the solution for the problem. And this has been proven in multiple people. Like, now you can identify a particular chemical, you can do a in silico model of that particular drug. Nowadays, this is happening even in a computer. And then you can even do simulations of what will happen to it in a computer, and then you can produce it and then take it to clinical trials and then give it to a pharmaceutical company to manufacture it. And nowadays, now this pharmaceutical company also is disappearing. You have this entire process happening within a hospital right next to the patient's bed. The introduction of CAR T therapy, if you have not heard so far, where they take the patient's blood, identify the T cells in that, and then they attach a particular genetic material to it, and it's given directly to the patient. So there is no more factory involved. You have the entire process happening right besides the bed of the patient. So that's how technology has changed. And that's all possible because of the bringing in of technology and better understanding of biology and better capabilities of being able to process this. And this is important for you all to remember also. You should know which focus area you should think about. You can't be sitting nowadays and think of a particular targeted therapy which still needs to go to the factory to get manufactured, because targeted therapy for a particular condition now has gone to the bedside. So when you are thinking of a solution, you should also keep in mind what is the current status of that particular problem and where are we heading towards, because you can't have a solution which will not be having any marketability in the future. So it is another very important angle of discovery. You can discover very good things, but if you're not able to make it get into the market because you have not stayed abreast with what's happening in the world, you'll also be at loss, because you'll spend a lot of money and resources, but you will not be able to really get that product into the market. So it's important to keep in mind that technology is moving so fast that it also has this kind of a danger of you becoming irrelevant by the time you come out with something. Okay, so drug discovery, when you're talking about from a targeted drug discovery, has generally has followed three kind of paths. You have a phenotypic screening. Basically, you look at drug molecules and you put it into a specific cancer cell model, and you understand whether that particular compound has something to do on the cancer cell. So this has led to a lot of cancer drug discoveries. Then you have a target based screening. You identify a particular gene or a receptor or something like that, and then you in a computer or in simulation, you kind of make a target that would go and attach itself on that particular target, and you see if it is able to kill that particular cell. Then you put it onto cell lines and you decide. And then now the latest one that is happening right now is the genomics assisted screening, which is happening where you are now going at gene level and we're trying to see if there's a defective gene, can we correct that defective gene. You have a gene editing that is happening, and that's the kind of new targeted therapies that are happening in drug discovery. So largely, a lot of the compounds have happened. If you ask me, the last 30 years have stuck in this target based screening. And because it's been quite easy to find ligands, and once you find a ligand which is a target, you kind of find something which will attach to the ligand, and then you can demonstrate that it has a cidal activity. So a lot of the antibiotics kind of developed like that. A lot of the antivirals are getting developed like that, and now we are looking at even cancer therapies also coming in this particular direction. But at the same time, there is also genomic assisted screening targeted therapies that are uh, if you ask me, phenotypic screening, it has more relevance from a disease part of it, because you can address a larger disease population, or you could even address a rare disease population. And you can go by that. What is the phenotypic expression? And then you can study the phenotype, and then you can go back. You will be having higher chances of finding a drug for that target, because the expression is like this, and so you'll be able to correct it. but it's difficult to form an assay for it and really understand the mechanism of action. And this is one of the reasons why Ayurvedic medicines and traditional medicine systems follow this path. If you ask me, they also have targeted therapy in their mind, but they have more of a phenotypic kind of a process. So they go directly to the disease and they'll study the disease process, and they say that I can change these disease processes by using this particular drugs. But you ask them to put it into an assay or give you an explanation of the mechanism of action, they'll find it very difficult to do so. And it addresses the issue about biological complexity. That's why they call it more kind of holistic medicine. And you can easily sample the entire population. You can kind of go out there and start looking at it. People will accept it. You talk about an herbal medicine for corona right now, people will just jump into it because it's explained simply. People understand, okay, you have a cold, you take something for cold, like take pepper for cold, your cold will disappear because pepper is able to kind of bring down the vascular dilatation that happens in your nasal region. And so you have an explanation for it, but you really wouldn't know. You ask exactly how pepper is acting on that, nobody will be able to give you an answer for that. Target based screening has poorer disease relevance because you go by a particular condition. You use, let's say, for example, penicillin. It was developed based on identifying certain bacteria which was getting stopped by this particular penicillin. So they thought that if you have that, penicillin was the considered to be the magic bullet, and so it said penicillin would cure all diseases. But then over time, people realize that you can't cure many infections with penicillin, and the bacteria also are able to kind of change its morphology. And so, similarly to that, also you start seeing in chronic diseases. You think you have targeted based therapy. I can give you an example: is a statin. What does statin do? Statin is a drug which lowers cholesterol, and it goes and targets the HMG CoA enzyme, which is a kind of a final step for the conversion of the triglycerides from your absorbed fatty acids. And they felt that that will kind of completely cure hypercholesteremia and reduce heart disease. But then you started understanding that some people were having problems because of that, because you do need some cholesterols to be produced. At the same time, it is not really reducing in everybody. So now people have moved into genomics, where we don't really understand the disease relevance because it's expensive. We don't know about druggability because it's huge. Because once you do a genome assay of a particular whole genome sequence of a person, the amount of data that is sitting in front of you is huge. But then you can, on using technology, you can easily identify a mechanism of action. You can address the biological complexity, and it's easier to do sampling of an entire protein because you've got the entire sitting in front of you. You can look at it from all angles. So it's always a balance between these three. So you really cannot take one particular target and take it forward because it will have something which will be there or something which will be absent. So the approach should be a balance of all three. You should be able to get a phenotypic expression for whom you can identify a target, and then look at it from a genetic perspective to see if that target is overexpressed or underexpressed in a particular disease population. So you can't go with just one path now. You have to bring in a combination of all these three parts to really have a successful targeted therapy to go forward. The other challenge that you have with chronic diseases is this latency. You say, okay, somebody's got exposure. Now you have heart, you may be having hypertension, you may be having diabetes. But will you die tomorrow? Will you have a heart attack tomorrow? No. You'll be fine for about 15, 20 years, or maybe even 30 years, you'll be fine. You will have no manifestation of that particular disease at all. And is asymptomatic in many people. So it takes time for the disease to kind of bring in a structural change in your body to start bringing about a form of a disease which will be very reactive. Now, if you ask me, COVID has kind of contracted this whole thing. You get exposed, and within 5 to 7 days' time, you have a very altered structure function happening, and then you have disease. But if you really ask me what is contracting it, it's not the virus. It is our existing latent forms of chronic diseases, which is actually created that, it's like, it's like a chemical reaction. You're kind of giving it a process to be able to, react much faster. Because always we are influenced by, and I think I shared this in the beginning, chronic disease is multifactorial. So you need to have a combination of multiple things to come on to you to really express yourself into a disease. Now, do you say that there's a big debate going on, use of mobile phones causes brain cancer? Now, you can't confidently say no, nor can you say yes. Because theoretically, yes, it may be possible because it's some form of radiation that's coming out of it. But it is the kind of number of hours of use that you have, what other factors are there in your body. So, like that, it's difficult to work when it comes to chronic diseases in multiple things. Diabetes, you look at hypertension, anything, any chronic disease you take, even chronic infectious diseases also. You see, some people have HIV, but they live kind of normal lives. Some of them have HIV and they kind of are struggling every day. And so there is always this multifactorial thing which is kind of influenced by the environment. I think we kind of covered that, and it's important for us to keep in mind that anything that we build is able to address a system level and not, that's why I was trying to say you can't go with just one path of drug discovery. So this is another example of how you have disease pathways and you have any compound acting on different parts of the disease pathway. So you can look at a particular receptor and say that I'll affect this receptor, but there may be something else that is also influencing the receptor. And this receptor may be also acting on something else, which is the important function. And so this was a kind of a schematic that we drew when we were looking at certain natural products and to see if how the natural products can act on different levels. So this is a kind of a systems biology. So if you look at a target, it needs to be not only acting on one level, but it should be also being able to influence. So if you're using something to influence this, you should also be able to influence G2 so that its influence on P2 is reduced. And so anything that happens from G1 will not affect it. So if you look at it from HMG-CoA reductase, if you just cut off HMG-CoA reductase, all cholesterol production cuts off. Now you do need HDL production. You do need certain types of lipids to be produced so that you have normal nerve function. And so that's why people start developing muscle pain. People start developing, muscle wasteness in that. So you can't go with the targeted therapy and hit really high. You can't go down very low. So you need to identify that right place where you need to make the modification. And that's one of the reasons why also I keep saying that we can't go with just one gene therapy or a ligand therapy or just a phenotypic therapy. You need to bring in all three components. So who started targeted therapy? I think it started off with this concept of Paul Ehrlich's magic bullet. He was the first one to kind of talk about receptors, and he kind of used terminologies which people felt was at that point in time they realized that he was ahead of the curve, so people really did not believe in what he was trying to say, but we have come a long way since then. And so early sidechain theory was what kind of stimulated the whole cancer chemotherapy drug discovery, and they started understanding receptors at a different level, and they started looking at what could be affected at a ligand level. So you change the ligand, So the whole concept of targeted therapy, I think, in a way, has evolved from Ehrlich's side chain theory. And this is the complexity of discovering a receptor target. And I think I shared this earlier also. So you have a disease biology. So you know what is happening phenotypically in the person. You could express that in a in vitro cell line. And from that cell line, you could study the gene expression of it. Now, with advanced data, once the gene expression has happened, you can identify what proteins are getting expressed. Now, with the cell line, you can also do immunohistochemistry and identify the protein. So now, if you have this loop completed, then you know that this disease biology is caused by this particular loop. So, or else you start with the DNA sequence. That means you got a sample, you just sequenced the DNA, you've done a genomic database, and you understood what are the proteomics that gets produced by that DNA. And then you go straight to a gene profiling, expression profiling, and then you look at protein expressions, and then you can do some treatment out there. Or you could do this path, disease biology, cell line, immunohistochemistry, protein expression, protein structure analysis, and receptor identification. Now, the problem is that if you follow any one particular path, you will be limited on other paths. So you need to have something called as a systems biology. You should be able to, once you identify a receptor, you should be able to backtrack itself to see what are the proteins that is expressing, what are the protein expression profiling that is happening, from which kind of genes is it being expressed, how can you detect it, because you need immunohistochemistry to be able to identify it. So you're basically, now what you're doing is you're not really looking at the disease to target it, you're also finding out mechanisms to identify the disease in the population. So it's not just important to find a treatment, but there's no point in finding a treatment for which you cannot find the right patient. So now, if you ask me, pharmaceutical industry has gone into this whole mode of companion diagnostics. So they release a new drug and they bring out companion diagnostics along with it, because they feel that only if a person expresses that particular protein should you be giving this drug. So you see, it's a limitation of a targeted therapy, but at the same time, it improves the efficacy. And you'll be able to really be able to target somebody who's really expressing this kind of genomics and proteomics. You would have found the right kind of treatment for that. So receptor targets is an important pathway, but it has to be systems biology driven and not taking one of those paths of the phenotypic expression or target based therapy or a gene therapy. So you need to kind of interact with all of them. So again, this is a different way of expressing it in a computer based simulation model, where you have these free databases of expressed genomes, and you can kind of arrive at this kind of a pooling, and you can do a heat map to understand where your particular target is going to be expressed, or a particular treatment, where is it going to get expressed. So, like I always tell, you can't go with just one, So you have dabigatran, which was a ligand-based targeted therapy, and dabigatran is used for treatment of atrial fibrillation, which is again a chronic condition. Patients can live for their entire life with atrial fibrillation. The problem is that atrial fibrillation leads to stroke, so you need to give a drug which reduces that stroke. And so it was a ligand based kind of a treatment, which is basically they identified the factor 10A, and whatever are the inhibitors around it, they were able to come out with a treatment for it. But it is not able to optimize the affinity. And so it has a lot of adverse drug. You need to look at it from an absorption, distribution, and metabolism perspective. It brings in a lot of challenges there, because even though it's targeted, it acts on almost all factor 10, and that can lead to a lot of bleeding problems. So you need to adjust the dose. You need to make sure the patient is having normal renal function, because if he's not having normal renal function, you'll accumulate a lot more of dabigatran, and that's going to lead to problems. Now, that was addressed when rivaroxaban came out, which is again another, it's a basic fragment of P1 site, which was, again, it's used in the same target population. Rivaroxaban is given to the same set of patients who are having atrial fibrillation. And they said that replace a basic fragment with a neutral fragment using structure-based approaches brings in a more favorable, so you don't have to worry about renal function here. Now, rivaroxaban, they said, dabigatran cannot be given with people who have poor renal function. Rivaroxaban can be given with poor renal function because I've replaced something there. So you've modified it, and you've said that I've created a structure-based approach. So a ligand-based has moved to a structure-based approach, and it seems to be acting in a more safer kind of a treatment. So now dabigatran needs to be given twice a day. Rivaroxaban can be given once a day. You don't have to worry about renal function. It can be given in patients even with poorer renal function. So you see, you can play around with even the target that you've identified based on what kind of pharmacokinetic rate. So you can't just say that I found a target and think that the drug will. But each of these drugs are billion-dollar drugs. Dabigatran has sold more than $1 billion, and rivaroxaban is also sold for many $1 billion. But what I'm trying to say is that the subtle changes in targets also is based on the pharmacological property of the drug. And just by making minor modifications, you can bring about a major change in the way the drug acts. And so this has led now recently, off late, people now go into this kind of a systems biology approach where they look at all the aspects of it and they see whether they want for low efficacy or do you want to look for high toxicity. And the ideal one would be a high efficacy with low toxicity. So getting that is not easy, but these are just simulations that people have been telling that if you bring in a systems biology approach, you will be able to achieve it faster. So this is what I was trying to say. You can't just take one path, but you need to take all three paths. This is just another expression of the same thing. So you'll be able to bring our understanding of the entire signaling pathway a little more better if you are able to take a systems approach. And this is possible nowadays with computer simulations. So if I want to kind of come to my concluding few slides, when we're talking about targeted therapy, we need to have, even though we know of what is a target, it needs to involve the system and a whole. And you need to be able to do a systems biology approach. So you have a target tissue, you identify its receptors, you find a drug which reaches this at each stage of the disease. It may be in a latent stage or it may be in an advanced stage, and you are able to make a change to the biology which affects the DNA so that the cell then becomes normal. So you start pushing yourself from a diseased cell into a healthy cell. And there are a lot of ways of doing it. People are advancing itself in development of formulations for targeted drug therapies. You're using small particles called nanoparticles to administer the drug so the drug can go and act specifically on the receptor, or whether it is intracellular or extracellular. You have drug ligand conjugates. You find a ligand, you find a conjugate where it kind of combines together. You develop it in an animal model and then you bring it into, so you test it in an animal model and then you bring it to human settings. Or you could have a receptor targeted nano carriers. Nowadays, they're using biological carriers. You're having virus taking in these particles because they're even more smaller than a nanoparticle. So the process is changing from a simple chemical agent, which is just given, and where the body decides what to do, how much it gets absorbed, how much it gets distributed, how much it gets metabolized, how much it gets excreted. It is now saying that I will control its absorption, I'll control its distribution, I will control its metabolism. and I will also control its excretion. So you're able to target it at each of these pharmacokinetic levels and being able to bring about a therapy or a change according to what the disease requires. But it's not easy. So you have cancer chemotherapy using receptor based therapies where it is sometimes it's easy to find it. So you have receptors like transferrin receptors, folate receptors, EGFR, and integrin receptors. This is the kind of a mainstay of receptor therapy based targeted therapies in cancer chemotherapy. A lot of people are working on this because many of the cancer cells almost always express these receptors, and they feel that if there is that expression, I can then target that and I can destroy the cancer cell. Also, there are some things like PSMA on prostate. So these are more biologically expressed. So you have a biomarker for prostate cancer cells, and then you have the HER2 receptors in breast cancer cells. So you can differentiate whether they're HER2 positive or HER2 negative, and then you can give more targeted therapy for these people. Also, it kind of gives you a very specific as receptor tissue expression, which also tells you whether the chemotherapy is going to be effective on it or not. So whether you'll be able to get a disease regression or not. So helping in understanding the expression of some of the receptors is not only good for targeted therapy, but is also good for triaging your patients or understanding who is going to recover, who is not going to recover. It's not only in cancer that you're using targeted therapy, you're using targeted therapy in various chronic diseases. Now people are studying a lot about the beta 2 adrenergic receptor and expressed in the lung, and they're trying to understand. Now I'm sure with corona coming in, AC2 receptors will become a mainstay of understanding, because a lot of people are beginning to understand how AC2 receptors work. You have the transferrin receptors, which are also there. Then people have tried to use insulin receptors because you want to cross the blood brain barrier. You want something to do with cancers in the brain or any diseases like Alzheimer's, which affects the brain. The biggest problem is the brain is protected by the blood brain barrier. So you need something which will go across the blood brain barrier. So people have tried to use the insulin receptors. Insulin receptors are able to cross the blood brain barrier easily, but it comes with its own challenges of hypoglycemia and other things. Then another place where they're using targeted therapy is in inflammation, and people have been trying a lot about it in rheumatoid arthritis and other arthropathies, like the alpha v beta 3 expressed in angiogenic vascular endothelium. So basically what is happening is there's some kind of vasculitis that is developing, and they're trying to understand if this is reducing that inflammation that is happening. You have cell adhesion molecules expressed in various inflammatory conditions. Basically, people have better understanding of ischemic reperfusion injury. So post cardiac surgery, post limb surgery, they're trying to see if you can, reduce the amount of reperfusion injury that can happen by understanding the expression of this particular molecules, so that you can reduce transplant rejections and can also reduce inflammatory diseases that affect the skin. A lot of it now going into cardiovascular system also, and they're basically identifying specific peptides or antibodies which kind of helps in delivery of the drug or even expression of the receptor. Then you have the receptor specific. You have knowledges of different expressions of receptors, but so these are the challenges with targeted questioning. You have, from a receptor specific challenge, you do know the differential expression of a particular receptor is not the same in every human being. So a beta 2 receptor in a particular human being will not be expressed the same way in another human being. So just by knowing that this receptor is not good enough, you also need to know the gene expression, which you can understand from a profiling of that particular individual, and then whether that receptor will actually have a specificity or not. Does it have an accessibility or not? Is the cellular uptake, and is the once you start attacking, you do see a situation where if you start targeting a particular receptor, the cell will just immediately change itself and reduce production of that particular receptor. Then you'll have resistance coming in immediately. So receptor-based challenges are, those because it is dependent on a biology, and the moment the cell sees that that particular receptor is getting blocked by something, it will reduce its expression, and that leads to a problem. You have ligand-specific challenges. The specificity and affinity of the ligands are always difficult to ascertain. You can only kind of guess it. You can't really say whether it is 100% binding. And even if it's 100% binding, the problem is if you bind something 100%, if it does not dissociate itself, then the drug is of no use. So it needs to attach itself, and it needs to bring about a conformational change also. So you can't have something having 100% affinity. You need to have something which is able to attach but also release itself. So it brings in its own challenges from a ligand-based approach for targeted drug delivery. Then you have carrier-specific challenge. Which is the carrier that you use? I was talking to you about insulin. So similarly, you are now looking at viral particles. Now, what if the virus gets activated on its own? So now people are trying to bring in the smallpox vaccine virus. They're trying to see if you can attach it with oncolytic material or oncolytic material and use it as an oncolytic virus, because seems to have a property to be able to access some of these cancer tissues. But the big problem is, what if that leads to an outbreak of smallpox? So carrier-specific challenges, be it nanoparticles, be it lysomes or liposomes that you're looking at, it comes with its own challenges with regards to its affinity, its ability to cross the cell membrane, and also where it is being used and aspects of it. So the homogeneity and purity aspects of the receptors is also important. I was telling you, the same issue with regards to whether you use it as a pro-drug or you should use it as an activated drug. You give it as a pro-drug, does it get activated inside the cell? So all these challenges are there when it comes when you're using a carrier instead of a direct ligand to go and attach itself to the so in conclusion, friends, I wanted to say that our focus of any drug development should address the whole aspect of health. And if we do not stay focused on this definition of health, any amount of drug discovery is not going to be of any benefit to the human population. And so, if you are developing new drug targets, or you are in the journey of developing targeted drug therapies, we need to keep ourselves informed about the whole aspect about how health is to be attained in the presence of the various factors which influence it. And it's a difficult but a very exciting journey, and I'm sure that's why we need younger minds who can think out of the box to be able to come and solve the problems of health that is being addressing human beings. So I'd like to stop there, and happy to ask any questions. And I hope everybody is awake after this talk, and looking forward to hearing any views from anyone. Thank you.
Thank you so much. I don't think anybody can sleep now. They will have a sleepless night because we were all working in 3 dimension and maximum to systems biology, but you have introduced a multi-dimensional space. So I think we should have sleepless nights now, Dr. Alban. Thank you so much. It was wonderful. Now, guys, please ask questions.
Excellent presentation, Dr. Alban. You've very nicely covered something that is so expansive in 1 hour. Great presentation.
Thank you. So we have some students who are part of the hackathon, right? Here.
We do have some. We have around 24 students who are participating in hackathon.
Okay. So what do they feel about their calling for getting into this hackathon? What brought them into participating in a hackathon for finding solutions to health? Anybody wants to answer that?
Varun, can you answer that?
Not really sure, ma'am.
Anybody else?
Hello, ma'am. Hello, sir. Good morning. I'm Sarah Philip, and my main intention of joining this hackathon was to actually expose myself to the problems in the healthcare sector, and also to make small baby steps, because this is where I see myself working and similar problems. So that's why I chose this. Uh, that's why I decided that I should maybe take part in this hackathon.
So Sarah Philip, what's your background?
Sir, I'm a second year biotechnology student in RV College of Engineering.
Okay. So, what have you understood so far about the merger of biology and technology in a very broad sense?
Sir, I think like traditional biologists used to have a sort of resistance towards uh, adopting maybe, this is my very basic understanding of it, had this sort of resistance towards adopting very revolutionary or new techniques. But now we all are really converging to this one point where we can't do without the other. So the the biologists cannot do without the latest technology, and of course without the proper conceptual knowledge and the mechanisms and the working of the body, the technology is also incomplete. So I think it's very important for it to happen.
Good. Yeah, because see, when this field of biotechnology was being introduced around the time when we were finishing, I was graduating, people really did not understand much about it. And they only thought at that time it was only meant for drug discovery. And people thought that you could become a biotechnologist and discover new molecules in biology, because that was a time when recombinant DNA was being used for manufacturing drugs, and they thought, okay, you need somebody with the biology knowledge to do something more technological. So, as you know, traditionally drug discovery has been the forte of chemists, and so they are the ones who kind of brought in this whole traditional from a traditional system of medicine to a modern system of medicine. So I think the introduction of biotechnologists, or the whole field of biotechnology, was to expose yourself into that part of it. When we studied botany and zoology and all that, it was very boring subject, but I'm sure now it's become very interesting subjects, because though there is now an application of those subjects, because you need to now have strong understanding of your botany and your zoology to be able to really understand the advanced topics of biotechnology. Good. Anybody else? I thought there were about 23 people participating in the hackathon.
Yes, sir. My name is Ganesh. This is the first time I'm entering a hackathon of this sort, and this is giving me a pathway to what really is drug designing, and it's background is, and yeah, I'm learning a lot.
What is your background? You're also a biotechnology student?
No, sir, I am an electronics and instrumentation.
Oh, good. So, what do you think is the role of an engineer in medicine?
Basically, what I think is diagnosing itself is a major part where technology comes in the initial stages, but uh, uh, I guess it has to be like more sophisticated. Like it has to be like improved a lot. So that's where our engineering, technology comes
Yeah. See, there's a difference between the way an engineer thinks and the way a doctor thinks or a biologist thinks. Any idea what that is?
in. No, sir.
Anybody else? Okay. See, a
Hello,
biologist. Yeah. Tell me, who is this?
I'm Sai Drishya.
Yes. Yes, I.
I'm giving like my guess about this. So, medicine-wise, they will think what medicine should be or what drug should be prescribed to solve a problem. Biologists will think of how the drugs can be prepared and what pathway should be taken. Whereas an engineer will think how the drug will be prepared in the shortest span and what other technologies can be involved to do so.
Yeah, that is going down into a little more specific, but to be a little more on the broader aspects. See, biologists always work on probabilities. Okay. And so they can only predict certain things by looking at a probability aspect of it. And they are dealing a lot more with associations and other things. Whereas an engineer, because your approach of in any engineering problem is to find the root cause, and you go directly to the root cause and you fix the root cause. Once the root cause is fixed, then you kind of consider the entire problem will correct itself. And this is something which is lacking in biology, and this is where I really see a merger of the two fields coming in is going to make a difference. The way engineering is taught and the way biology is taught, it needs to kind of come together to find this common ground where the biologist will explain the association of different factors and the complexity of biology, but the engineer is able to break it down into smaller parts and say, I will come down to the root cause and I'll address the root cause. And this is where systems biology will happen. And so you need this convergence of these two fields to come in to really solve health problems of the future. It can be in drug discovery, it could be in healthcare delivery, it could be anything. Understand?
Yes, sir, that makes sense.
Okay.
So it's me, Sarah, again. Actually, I wanted to ask. I was reading a recent paper from IISc, and they were mentioning that they would isolate cancer cells from a patient and grow it on a scaffold and test all possible drugs on it. And this was done for, I think, breast cancer, and they would come up um with the best suitable drug for treating a patient. So, to what extent is this effective, and uh, how much is how much is the success rate as per your knowledge, sir? Because it would take 10 days to grow it on a scaffold, and we are only testing already existing drugs on it.
Yeah, so I think I covered this component with when I said the genome based approach and the ligand-based approach. So what you're told is just one component of it, where you, instead of, it's a little advanced form of doing cell line and cell culture. So it's called an organoid. So basically what happens is, see, when you get a cancer cell from a cancer tissue and you culture it on a cell media and you grow it, that particular cancer cell does not express all the biology around it because it's sitting in a completely homogeneous environment there. And so the amount of receptors it produces, the amount of certain ligand expressions it does will be different compared to giving the same environment which is there within the body or within the organ in general. So the concept is now trying to replicate how the cancer sits inside the human body by doing an organoid. Now, if you ask me from a time perspective, yes, it will take time, but there are now culture medias which are able to kind of create that much faster. But nobody gets into that without first doing a kind of a computer assisted animation. And even that is not foolproof evidence because you need to replicate that in a closed biological system. So it has to be expressed back inside in an animal model to say that it works in that direction. So it is only a starting step, but you will be in a much better position because if you've just identified a cancer cell and you find that a particular receptor and you give it something and you see the cancer cell dying, it doesn't mean that the same cancer cell, when it is existing in a human body, will express the same set of receptors. Like I said, receptor expression would be a big challenge there. So that is what I was talking to you about, the challenges with looking at a receptor expression. So that challenge is kind of addressed a little better when you are building it on an organoid, where you kind of create the environment around the cell, which is kind of replicating the thing. But still only the starting step. You still need to put that and demonstrate it in a closed biological system like an animal model, and then take it into a human being because you need to establish its safety there before you put it into a human being. And so this is kind of getting replaced a little bit with the introduction of CAR-T therapy, where you've identified this particular aspect that you are able to express a particular ligand when a particular tissue and in an organoid, and you know if you target it, you're able to bring it out. Then you take the same in CAR-T therapy. What it does is that they use the patient's own T cells as the drug delivery system. So you have killer T cells in your body, which your body produces. Currently, may not be having sufficient immunogenicity against that particular cancer cell. You train that, and then you send it back into the system. So that's where this kind of research that's happening is really moving concepts in that direction.
So hello, sir. My
Yes.
name is Varun. So I'm actually interested in machine learning, so especially deep learning. And in the past, like I think 3 months ago, so I made a deep learning model which was able to generate new drugs, but based on pre-existing drugs. So it uses something called a variational autoencoder, which is basically a generative. So usually machine learning models are used for classifying, but machine learning and deep learning models. But variation autoencoder is a deep learning model which is used as a generative tool. So I used it to make some, input some pre-existing drugs. So it by converting it into smile strings and outputting new node. And so I also used a classification system based on deep learning to figure out if the drug is active or not. And I used, I think, various parameters such as log P, molecular weight, etc. So the problem with this method is that you actually need a large number of pre-existing molecules to actually get new ones, right? So do you know any new technique or a machine learning technique which can actually generate drugs without the need of pre-existing compounds for that drug, for that biological target?
First of all, your audio wasn't very clear, so I've not been able to completely understand your question. Secondly, I am not an expert in machine learning. I can give you a very basic understanding about this, is that machine learning and algorithms are being used extensively in drug development. And that's been one of the mainstays of drug discovery or repurposing that's happening with COVID-19 and heavily used in cancer drug discovery. People have been able to use known compounds which have been discarded because of its toxicity profiles and unable to kind of reach the market because it cannot sustain itself from a regulatory standpoint of view. These molecules are now being studied using artificial intelligence models, and they are trying to see if they can bring that drug in a different way, in a different dose, or more targeted therapy to be able to be used in some disease where it was no. So probably they found a particular drug which was initially developed as an anti-cancer drug, but then they found it has another property of reducing cholesterol or maybe reducing something else. So that is what is um, uh, kind of emerging right now because of artificial intelligence. Like for example, there's a drug called niclosamide which is used as an anti-helminthic, but artificial intelligence models have shown that it is a very effective drug against COVID-19, and it can actually reduce the cytokine storm that is caused by COVID-19. So people have now started doing clinical trials using that. Another drug which was discarded by everybody is thalidomide. Thalidomide because it caused phacomelia in pregnant when pregnant mothers took it. It was given as a drug for pain relief and other things, and as an anti-emetic drug for pregnant mothers, and it caused that phacomelia, which is loss of the four limbs being developed. But now that drug is being repurposed right now in certain chronic conditions, and they're thinking that if we can deliver it more safely in certain conditions, it has a much more effective drug. It is being looked upon in certain types of pulmonary hypertension and other forms of congenital diseases. So, like that, for example, artificial intelligence is throwing a lot of light on this because it's a model which will keep learning. So, you can feed in all the data that you have from these different sources and make the machine learn. And but the problem there, one of the challenges there, and that's why I brought in that slide there, the initial step that you go is go with a correlation, a signal processing of a correlation. And so you should be very careful about that. And that signal processing using a correlation, if it is not biologically explained, then you are not going to go far. So a machine learning algorithm per se, because entire machine learning is all based on correlations, and it tries to pick up a particular signal and tries to see if that signal explains a particular effect. And so the problem there is this association and causations don't play hand in hand, and so that is why I was telling you about being to be careful about correlation.
Varun, if I may,
Yes,
a few thoughts here. I think machine learning, AI have come in where the problems revolve around a lot of data, right, which the human brain cannot comprehend or interpret very easily. So essentially, computationally, you're able to essentially detect signals that you would otherwise not be able to see, either as a biologist or a genomic specialist. So this is, for example, let me give you a problem. I'm not sure how many of you have heard of liquid biopsies, right? Circulating tumor DNA, which is a very, very small portion of DNA that's floating around in your blood or plasma. And now this is used for a few things. One is monitoring treatment response, right? So you essentially know the mutation profile of the tumor you had, and then post chemotherapy or whatever treatment you're going through, you draw your blood over a period of time and see if you can still detect those mutations in your blood, because they represent a small fraction of your circulating tumor DNA. Typically, this is done either by PCR or when you do sequencing. The problem you face is that you have so much data and you're looking at very, very low frequency changes. And when you don't know the mutation, you're looking at a lot of background noise. Like typically any human being you take and you align to a reference genome, you end up with a million variants. Which of those are relevant? What are the patterns that you see and how consistent are these? These are typical problems that machine learning algorithms work on and help us to resolve and come up with signatures or biomarkers that can help us, associate them with a certain biology or significance for the disease. I think that's in a nutshell, typical applications of machine learning. Also, AI has helped a lot in radiology. For example, I think it's, as Dr. Albin was mentioning, in COVID, for example, they're looking at, the X-rays of lung and seeing if one can interpret or diagnose the disease very early on, which a human eye cannot detect. Right, so machines can be more sensitive because you're pixelating. You can pick up even finer and high resolution images that can help interpret much better than human eye can. Another classic example is, I think, what Arvind at Madurai did on being able to detect diabetic retinopathy much earlier and better than a doctor can just by looking at images of the retina. So these are some places where these applications are very, very valuable and almost threaten to replace humans in terms of their efficiency and sensitivity.
Yeah. So the reason why I started, dwelling into machine learning and artificial intelligence is because I read somewhere in a book that in the process of generating new drugs, usually what happens is scientists or a group of people who have a lot of knowledge in the subject of drug discovery, etc., actually sit together and make small perturbations to pre-existing compounds and test if those new compounds can be is effective in the real world scenario. So I thought, oh, why not use an AI model which can do that for you? But the problem is you end up generating a lot of drugs which, like you said, toxicity, and it may not be viable in the real world scenarios. So maybe if we can combine the knowledge of humans in a basically a knowledge driven approach with the power of AI, we could actually make huge strides in drug discovery and de novo drug generation.
I don't know how many of you know it. It's about a couple of $2 billion before a drug is in the market. And as Dr. Albin showed earlier in his presentation, it's a multi-step process, right? Spanning 8 to 10 years, requiring various specializations. So any contribution to any of these steps could accelerate, right? So let's say if it takes 10 years to do something, clinical trials being a significant part of it, which is towards the later stage of drug discovery and implementation. Earlier on in the discovery process itself, very early on, which targets, how do they work, what pathways, what dose, administration. So there are multiple steps, and many of these tools and methods can accelerate any of these steps, right? It's not necessary that one solution can essentially take you from a problem to a drug like magic. I don't think that's feasible anywhere soon, but definitely these things help accelerate. So maybe if something will take you 10 years, it would bring it down to 6 years by reducing the failure, by being able to better predict the outcomes, or by reducing the time to do that analysis. Right? These are two critical ways that one can accelerate. And I think AI will definitely contribute to those steps in the drug discovery process.
Yes.
Sir, myself, Shilpa.
Thank you, Dr. Vidya. But yes.
I say myself Shilpa. So now we have so many advancements in biological discoveries.
But it is not audible.
Hello, is it audible now?
Yes, now it is audible.
Yeah. So now we have so many advancement in biological discoveries and technology. So we even have genome sequencing with which we can understand exact locus of a gene and many more knowledge about particular gene. So with already existing technology, is there any way we can stop mutations? Because cancer is mostly caused due to mutation in a healthy cell. Is there any way like we can stop or prevent a mutation from like the cell from mutating?
You will get a Nobel Prize if you can find that out.
I know that, sir, but is there any studies going on?
So, see, the thing about biology is adaptation. Okay. So, mutation happens because of adaptation. I don't like to call cancer a disease. I call it's more of an wrongly expressed physiology. Because what you find in a cancer is actually your own tissue. It's your own, biology which you have been born with. It's just that it's sitting at the wrong place at the wrong time. And you do have your own systems to be able to. So let's understand mutation. The mutation happens because of stress. And if your body is functioning at a particular optimal level, uh, and you have your genes coding on a regular basis, it's like a production line. Whenever you have a production of a particular type of protein, that protein is going to stop. It's going to make some errors in its coding, and you're going to have these changed proteins coming out. So the body has its own mechanism to correct it, and it also goes by the amount of expression of that particular error happens. If that error happens too frequently or in larger numbers. So, current understanding of science and biology, we would say that mutation cannot be stopped, because unless the cell dies, the cell will continue to divide, and as long as cell divides, there's going to be, uh, the expression of its proteins, and once that happens, there is bound to be an error in the expression, and that leads to a mutation. And then if the cell is stressed, it will change. So it will stop uh, producing certain proteins, and so because it tries to go into dormancy, or it will try to change its biological pathway which is being done, because it needs to survive. A mutation is kind of a survival tool of a cell. So if you ask me, can we block mutation? I don't know. I'm not saying it's not possible, because we don't know what people will find in the future. But in our current understanding, mutation is kind of an adaptation process, and every cell needs that kind of a process. Like I told you in the beginning, now if you take the genes of all of us who are like now are forced to sit at home, I'm sure we'll be producing some different types of proteins, or many of our cells would have mutated by now. And I'm sure, in a very simplistic way to put it across, I'm sure many of you youngsters will be less irritated with your parents now, because you've been living with them for the last 3-4 months, and you are dependent on them. So teenagers, especially adolescent age group, they don't like to be around their parents, but now you don't have a choice. And sure, there is some kind of an expression of a protein which is mutated within you, which is making you less irritated or more tolerant to your parents. So that way, if you look at it, mutation is something which is very biological, and it keeps happening. What needs to be done from a cell part of it is trying to understand, at least this is what I feel, that if there is a mechanism to measure your biological activity at a particular time, at any given point in time, is your expression of your proteins, is your expression of your certain components, which are, it may be an electrical signal, it may be a biological signal. if that is a measure that we can find out and we see that there is some overexpression or underexpression of a particular gene, then maybe we can try to find answers to some of the questions that we have with regards to cancer. So really speaking, I don't believe we're going to have a cure for cancer. And that is something which people should stop pursuing immediately, is what I feel. Believe that you need to be able to detect cancer early, because the best treatment for cancer is surgery, except for liquid cancers. But there also you can do a procedure like bone marrow transplant, but which is again a surgical procedure, which has involved. But any organ-based cancer, your best option for cure is surgery. So if you can pick up a breast cancer or an ovarian cancer or a lung cancer or a liver cancer, sadly, nowadays even with brain cancer also you can surgically remove it when it is early. So that is the only cure that you can come out with for cancer. Now, mutation, if you ask me, is a very broad field. It's the big problem that you see with your microorganisms also. You see it with your antibiotics. So if you ask me if there is a application of understanding this mutation, it's definitely there. But it's more about trying to understand about mutation and trying to see if we can pick up mutation rather than trying to stop mutation, is what I think at this point in time would be a better uh, challenge to work with. Okay. So I think we're done for the day. What are your next session?
If you have any questions, something where they can work on hackathon, you can post that and I will stop streaming YouTube. So this is exclusively for only these participants, which they will give us some answers and the best answer will be divided by Dr. Sudha Rao.
Okay. You mean I can suggest something?
Yes, as for the students to work on and get with a solution, something which they have